Hello, I am Ajay Tijore. I've been working as an assistant professor in the Department of Bioengineering for the last 3.5 years. I did my PhD from NTU Singapore in stem cell bioengineering field. Then I completed my postdoc from Mechanobiology Institute, National University of Singapore in cancer mechanobiology field. There I worked with Professor Michael Sheets as a postdoc, and we discovered that the physiologically driven mechanical forces can be used to selectively kill different types of cancer cells without damaging normal cells. Moving ahead, we also developed a non-invasive and a novel ultrasound-based technology to selectively kill the tumors grown in mouse models and inhibit their growth. Based on this expertise, I established my mechanobiologics lab at the Department of Bioengineering in mid-2022. Currently, the lab focused on two major themes. The first is the cancer mechanobiology, and second is the stem cell bioengineering. In the research in the cancer mechanobiology is focused on recapitulating the architecture of tumor microenvironment using the microfluidic devices and the bioengineering platforms. And the next objective is to study the effect of this ultrasound-mediated mechanical forces on cancer cells grown on these different platforms. In particular, we are interested in oral cancer cells because oral cancer is the second most prevalent cancer in Indian society. The outcome of this study will help us to develop a novel ultrasound-based technology for the oral cancer treatment. The research in stem cell bioengineering is focused on optimizing this ultrasound-mediated mechanical forces to regulate the stem cell differentiation. In particular, we are interested in regulating the stem cell differentiation, or I would say the inducing the stem cell differentiation into the osteogenic lineage commitment. Ultimately, this will help us to develop the 3D osteogenic graft for the clinical treatment and the regenerative purposes. Thank you.
Hello everyone, I'm Alka Kumari, 4th year PhD student at Mechanobiologics Lab, Department of Bioengineering. I work in cancer cell mechanobiology, where as we know that when cancer cell migrate during the invasion one place to another place, it uses the tumor microenvironment area, and in this area confinements, very narrow 3D like channels are present. So my project is to check the role of these confined spaces on mechanical force induced cancer cell killing. Here we are using ultrasound, low frequency ultrasound as a mechanical force. I am going to talk about how the confinement suppresses the mechanical force induced cancer cell killing. So for that, I made a microfluidic device. In that, we let the cells, cancer cells pass through those channels, and we treat it with the ultrasound, and we check the behavior and what is happening to the apoptosis. And we found out that confinement suppresses the mechanical force induced cancer cell death. So what we saw that when cells are in unconfined state and when we give the ultrasound forces, there is an increase in myosin 2A contractility, Piaggio 1 focal adhesion colocalization, and there is an influx of calcium that induce the apoptosis in cancer cells. So when the cells are in confined state, squeezed in a 3D channel like a structure, and we give the ultrasound forces, there is a loss of focal adhesion, the attachment of the cells to the substrate, and they are not able to sense these mechanical forces, and there is no increase in myosin 2A contractility and the apoptotic pathway induction. So these cells, these confined cells, are unable to feel the forces that we are giving, and then they don't die. They inhibit or suppress the mechanical force induced cancer cell apoptosis.
Hi, I'm Akshay from Dept lab. I'm a collaborative researcher. So we've been collaborating with Ajay's lab for a very long time now. So we have developed this bioreactor from which we can control the frequency and the mechanical forces that cells are going to experience. So we have made different type of bioreactors. Some of them are used for live cell imaging, and some can accommodate different kind of tissue culture plates. So one of them I can show you now. It is in incubator. So yeah. With this bioreactor, we can treat several samples simultaneously. Thank you.
Hi, I'm Anaswida, a fourth-year PhD student at the mechanobiology lab. Herein, we use low-frequency ultrasound to selectively take cancer cells and not the normal ones. So, my work basically is to understand how the extracellular matrix nanoscale ligand spacing affects the ultrasound-mediated cancer cell killing, or the mechanoptosis. Now, what is nanoscale ligand spacing, and why are we asking this question? We know that the adherent cells bind to the extracellular matrix fibers via the binding domains present on them, uh, contacting the cell surface receptors by the integrins. Now, these binding domains are known to be regularly interspaced at nanometer length scales, which is characteristic to that, and changes in which have several implications on cellular behavior in terms of their morphology, their ability to migrate, their survival in presence of chemotherapy drugs, etc. Now, these cellular behavioral modifications are happening due to the modulations in myosin contractility, which is also known to play a key role in driving the mechanoptosis activated by ultrasound treatment. For our studies, we use fabricated gold nanodot platforms. We culture cells on very nanoscale ligand spacing, treat them with ultrasound, and so the observations. What we have discovered interestingly is that the optimal ligand spacing, which is known to support for a stable focal adhesion formation, cell spreading, and increased contractility of the cell, also promotes the mechanoptosis process driven by ultrasound treatment. For our study, we also have taken physiologically relevant optimal spacing and suboptimal ligand spacing because in diseases like cancer, wherein fibrosis is known to happen, it increases the ECM deposition around the tumor microenvironment, increasing the ligand density, which is why we chose a lesser ligand spacing compared to the optimal one. And we found that the optimal ligand spacing promotes the ultrasound-mediated cancer cells, while the suboptimal spacing doesn't. Interestingly, when we combined the suboptimal ligand spacing, we treated that with an integrin blocker drug known as silengitide and then did the ultrasound treatment. We were able to show synergistic effects of the mechanoptosis and were able to see an increased cancer cell killing on these ligand spacing as well, paving a new way of combining the integrin blocker drugs along with ultrasound to show an increased mechanoptosis.
Hi, I'm Rashmita Luha, a third-year PhD student under the supervision of Dr. Ajay Tijore at the Mechanobiologics lab. I'm currently working on developing a non-invasive oral cancer therapy using low-frequency ultrasound. Recent studies have shown that low-frequency ultrasound selectively kills cancer cells without damaging normal cells. The motivation of using this ultrasound-mediated mechanical forces comes from the growing understanding of how mechanical forces are involved in cancer progression, and this has opened up possibility of using mechanical forces for cancer treatment. According to GloboCan 2024, oral cancer in India is the second most prevalent cancer owing to the heavy consumption of tobacco products, areca nut, alcohol, and smoking. The most interesting part of my project is that I'm isolating a number of patient-derived oral cancer cells, and we are in collaboration with the Ramaya Memorial Hospitals. We are treating these patient-derived oral cancer cells using a custom-based ultrasound setup, and we are optimizing the parameters so as to get the maximum killing in these patient-derived oral cancer cells. I am further working on developing a microfluidic channel which can mimic the tumor organization of the oral cancer, and I am checking the ultrasound effect on it. In future, we are planning to validate the ultrasound effect in vivo mice model. This project incorporates the heterogeneity of patient samples and shows how low-frequency ultrasound selectively induces apoptosis or cell death in patient-derived oral cancer cells. This approach suggests a possibility to lay the foundation of mechanotreatment for oral cancer, which will be personalized, safer and non-invasive.